Thank you, Krista. And thanks to the organizers of this event. Um, So I'll be talking today about our lab's work on molecular chaperones and their role in pink-1 Parkin mitophagy. So Amy did an excellent job with the overview for Parkinson's and even some of the proteins that I'll be talking about. So uh, just a really brief overview. Parkinson's is a neurodegenerative condition that's mostly characterized by two main facets: protein aggregation and mitochondrial dysfunction. Um, and we believe both of these are linked, and we believe that molecular chaperones can play an important role in regulating both of these processes. So with mitochondrial dysfunction in particular, mutations in genes, including PINK1 and Parkin, are known causes of Parkinson's disease. And both of these proteins are very interesting because they have defined roles in the removal of damaged mitochondria in a process known as mitophagy or mitochondrial autophagy. So we hypothesized that molecular chaperones and co-chaperones regulate mitophagy and protein aggregation, acting as critical hubs that can potentially be used as targets for therapeutic interventions. So I just have a schematic at the bottom here. So under baseline conditions, you don't have, you have Parkin that's localized to the cytosol, and you have PINK1 that's generally degraded. So under mitochondrial depolarization, mitochondria becomes damaged, the pink one gets stabilized on the mitochondria, and the Parkin ends up, the Parkin, which is a E3 ligase, ends up getting or localized to the mitochondrial outer membrane, where where it functions as a ligase to ubiquitinate a lot of proteins. Pink one can it acts as a kinase to phosphorylate a lot of these substrates. And what happens is that this generates cycle where you have the autophagy system activated, which causes the autophagosome production or the formation around the mitochondria, which sends it for degradation. So this is a very rough idea of what's known about the PINK1 Parkin pathway. And in 2019, we published a paper that showed that BAG5, which is a co-chaperone of HSP70, can inhibit key components of this. So it can inhibit the translocation of Parkin, and it can also um, alter the functions of Parkin such that it promotes the cell death pathway and inhibit mitophagy, which is generally considered a protective pathway, the removal of damaged mitochondria. So we wanted to look at this a little bit more closely. So so most of this work involves sort of cell culture-based systems. So we have these U2OS cells, which we overexpress GFP Parkin to be able to see it, and it has an MT camera reporter, which is a pH-sensitive dual excitation reporter. So under certain conditions, it excites at a certain wavelength. At a pH that's below 4, it can excite at a different wavelength. So we use this to be able to look at this very dynamic process. So using these cells, we can get some idea about Parkin recruitment, levels of mitophagy, and we can also use this tool to look at cell viability with other markers. Um, so with this as a background, we wanted to see what are the other substrates involved in this pathway. So using iTrack mass spec with the Smith-Holmes lab at UFT, we were able to identify CHIP and a phosphomutant of CHIP as particularly involved in this pathway. So we wanted to know a little bit more about what these proteins are doing in this pathway. So in addition to using some of the tools that we had, including the cell-based systems, we ultimately found that CHIP is another negative regulator of PINK1 Parkin. So in addition to the cell-based system, we wanted to see whether we could look at other tools, so more of an in vivo system. So using some mitophagy reporters that have been generated in worms, we perform crosses with animals that are lacking CHIP. And what we found in this case was that CHIP negatively regulated PINK1 Parkin mitophagy in neurons. And so this provided a little bit more evidence to support that BAG5, along with CHIP, both of these, which are HSP70 co-chaperones, can inhibit PINK1 Parkin mitophagy in not just cells, but also in neurons as well. And then we also wanted to see whether we could try to see this in a PD-related model. So in our lab, we use the rat model of Parkinson's disease that's caused by AAV overexpression of A53T. So we initially ran this as a pilot study just to see, okay, what can we see? What can we say about Parkin, pink-1 Parkin mitophagy in the A53 system? And it was actually very interesting that, so the rat model is a 6-week model. So by the end of 6 weeks, they show progressive behavioral deficits and also a lot of TH loss, which is one of the characteristic signs of Parkinson's disease. So we noticed that at a very early time point, at 1 week, before they had any TH loss, that we could see a very strong induction of pink-1 Parkin mitophagy with the A53T overexpression. This subsided later on at the 3-week time point when we did have some TH loss. So this seemed like it's a very sort of early prodromal step in sort of the effects that synuclein might play in Parkinson's. So the work that we're doing now is trying to characterize the effects of CHIP on the system. So this is just very preliminary data, but it does seem that when we overexpress, when we co-inject CHIP along with A53T mutant synuclein, that we can see a potential reduction of that increase in mitophagy that we saw. So this is still preliminary. We're still putting together the paper here, but it's interesting to show that both HSP70, co-chaperones in BAG5 and CHIP may have a role in this. And it's sort of an interesting link because regularly molecular co-chaperones are just tend to be thought of as involved in protein folding, protein misfolding, and protein degradation, but all of these systems are in play and related to mitophagy as well. So the next steps for this work is to include some of the phospho-CHIP mutants we've also been studying, and ultimately to even see whether this can be relevant in a human PD model. So looking at organoids that have mutant alpha-synuclein expression as well. So I'd like to thank the Kalia labs, Dr. Sunil Kalia and Dr. Lorraine Kalia, and I'd also like to thank MITO2i for supporting a lot of this work that we've done. Thank you.